Hello, I'm Dan Tempest, and today I'm going to talk to you about ion channels and neuronal communication. But to start, I just want to talk about some general cell communication. So, in general cell communication, again, this is how cells are going to communicate. So, it all starts here with a signal molecule, also known as a ligand. So, the signal molecule will bind to a receptor. Now, this specific receptor you can see is on the cell membrane. It's not always the case. Receptors can be intracellular too. It just kind of depends on the characteristics of that ligand. So if we have a hydrophobic nonpolar, or maybe say a steroid hormone that can pass through that cell membrane, we could maybe then have an intracellular receptor. But if you have a polar signal molecule, then that molecule is not going to freely pass through the cell membrane, and it's going to have an extracellular receptor, which is what you see in this picture. That's going to happen. So we have to have reception. So the signal molecule binds to its receptor, which initiates the process. And without that, nothing after this can happen. So we have to have reception. And that's going to lead us to the second step, which is transduction. And in transduction, what we have in the model here, it says relay of molecules causing a signal transduction pathway. A lot of times this can include a phosphorylation cascade where proteins are turned on by being phosphorylated and turned off by being dephosphorylated, and that can transfer or relay that message throughout the cell. Oftentimes, there's also secondary messengers. Today, we're going to look at calcium as a potential secondary messenger, but cyclic AMP is another good example of a secondary messenger. And oftentimes, throughout this transduction pathway, um, you're also going to have amplification. So the signal, we can have just a little bit of signal molecule, and we can have huge responses to those signal molecules by having an amplification step here in the transduction. And that all leads us to the response. The transduction gets us to wherever we need to go in a cell to elicit this cellular response. Could be any number of things. An example of a cellular response could be a change in gene expression, but it doesn't have to be that. It could be, again, any number of things. All right, so today though our focus is on ion channels, and ion channels are often specific. They're not exclusively specific, but oftentimes they are. Ligand-gated ion channels is our first one here, upper left, and a ligand or ligand. As here we have a neurotransmitter as our ligand, and you'll notice that it binds to our protein. And the response then of that protein is to open, and in this case, it's allowing sodium to enter the cell. And you can see here, if we don't have a neurotransmitter or a ligand bound to that protein, it is shut, and sodium is not allowed in. So that's the ligand-gated ion channel, really common, especially in like synaptic signaling, where this neurotransmitter would be sent across the synaptic cleft and into the neuron, into the synaptic cleft to bind to a ligand-gated ion channel and potentially elicit a depolarization. Could be a hyperpolarization as well, depending on if it's an inhibitory or excitatory neurotransmitter. So that's the ligand-gated ion channel. We also have a mechanical gated ion channel, sometimes also called a stretch gated ion channel. The key here is that we have pressure against the membrane, and that pressure is what's stimulating the membrane protein to be opened. So you can see in this case, the ion is able to move in response to the pressure opening that membrane protein. These ones tend to be a little bit less specific. But again, if we don't have that pressure, the gate is closed, and so that's that mechanically gated ion channel. We can also have always open down here, lower left, and these you can see are specific. Here we have a potassium ion channel, and here we have a sodium ion channel. But again, as the name implies, they are always open. And then the last one down here, we have a voltage-gated ion channel, which responds to changes in membrane potential. So which there is a voltage across the membrane, which is the membrane potential. So in this case, we have sodium crossing in response to an open channel. So at a certain membrane potential, a voltage would be achieved, and that could open this ion channel. We'll look at that with an action potential here in a minute. but without that voltage being achieved, there is a gate that blocks that ion's passage through the channel. So those are the 4 different types of ion channel. All right, so the membrane potential has to be regulated, and that would be done by ion channels, but also ion pumps. An example of an ion pump would be a sodium potassium pump. Very, very important in a neuron. Its chief job is to regulate and re-achieve at resting potential. So, after following an action potential, we would have sodium and potassium on the opposite side of the membrane where they would be at resting. And in order to get it back to resting potential, the sodium potassium pump would be used to do that. And the movement of ions across that membrane changes the voltage across it. And we measure that by measuring the membrane potential. So that voltage is the membrane potential. And then due to the position of the ions, the cell tends to be negative relative to the outside. So we're measuring the inside of the cell when we're measuring the membrane potential, and it tends to be negative except for at the peak of an action potential. But at resting, it is negative. All right, so I'm going to go through the steps here of an action potential. I'll move my thing up here so I can see. So we all start down here at number 1, and this is resting potential. So at the rest, you can see all these gates are closed. Here we have a sodium ion channel, gated ion channel, closed, and also closed. Now, it's not directly told to us in this if that is a voltage gated ion channel or a ligand gated ion channel. I will get into that a little bit here as this goes on. Based on the imagery here, I would predict that this one is potentially a ligand-gated ion channel responding maybe to a neurotransmitter. Could be also a voltage-gated ion channel. It's not directly noted. And then here we have maybe a voltage-gated ion channel. Again, both of them could be involved. The specific action potential when it's going down an axon, it's likely a voltage-gated ion channel. But if when we have this initial depolarization, it could be on the axon hillock or at a dendrite being caused by a neurotransmitter. So if we go again down here to the resting potential on the graph, we would be right here at number 1. And again, all the gates are closed. Key thing where the ions currently are, we have sodium on the outside of the cell and potassium on the inside of the cell. All right, so now when we get to number 2, we have a depolarization. So a depolarization is a stimulus. In this case, it's an excitatory stimulus that's causing us to go towards threshold. And the reason why we're going towards threshold, which is an increase in membrane potential, is we have a positive ion sodium becoming into the cell. Again, that could be caused by a depolarization. A depolarization could be caused by a neurotransmitter, could be caused by a neighboring action potential that just happened and changed the voltage of the membrane. It depends on where you're at on the neuron. All right, so in this case, though, we reached threshold. So threshold is very important. So an action potential is an all or none response. If we reach threshold, it will occur. If we do not reach threshold, it will not occur. And so once that depolarization reaches threshold, we reach number 3, and that's the steep incline here. We call it the rising phase of the action potential. And you'll notice that all the sodium gates are now open in the imagery. So sodium is now rushing into the cell. And again, it makes sense why the graph would go up during this phase because we have a positive ion that is moving into the cell. And if we're measuring the inside, it does make sense that it would rise. So all those sodium gates are now open and sodium is flooding into the cell. Eventually, though, we reach the peak of the graph. And you'll notice over here in this image, we now have the sodium gates closed and sodium's passage is now being prevented. And now we reach stage 4, which is the falling phase of the action potential. And what you'll notice is rather than sodium moving, we have potassium leaving the cell. Potassium's gates are now open and potassium leaves the cell. And it would make sense. Again, a positive ion leaving the cell. Positive ion leaving the cell causes that graph to come back down. We actually even reach um, a undershoot, it's called, and that would be due to before that potassium gate actually closes, we fall below resting potential. And this actually also is associated with the refractory period. And I'll get to that here in a minute. But again, positive ion leaving the cell and potassium would be why we fall on that action potential. Eventually, all those gates would close, potassium's movement would also stop, and we would have to have the sodium potassium pump move those ions back to where they were at the start here at the resting. So we have to get back to resting eventually. The refractory period is important, and I'll get into that in a couple slides here where it's really important though in allowing the message to only be sent in one direction. So that's an action potential and how the specific ion channels, in this case gated ion channels, voltage, mostly ion channels down an axon, would propagate that action potential down a neuron. In my class, I like to use a simulation to model this. So the class would draw a neuron membrane, and it would use toothpicks for their gated ion channels. So the toothpick is open or closed and allows ion movement or not. The ions are simulated by different colors beads. So I would tell them to start at resting, and they should show me that sodium is outside, potassium is inside, and all the gates are closed. Then we move into that, depending on what phase of the action potential I want them to model, but we can move those ions and open those gates as we can. It allows me to really allow them to see it visually, which is really important for a lot of students. All right, so this is that slide I mentioned that I was going to have on. Oops. I'm trying to move here. With our refractory period. So the blue, as you can see, is what the part of the neuron that is achieving an action potential, and the action potential is moving in this direction, as you'll notice as we move down this image. The green part, however, is during the refractory period. So the refractory period is during that falling phase, as well as in the part where we move it back to resting potential. And during that time, another action potential cannot be sent in that area, and that allows the action potential to only travel in one direction, which is very important. And so after this action potential is achieved here, it actually triggers the next part of the neuron so it propagates itself all the way down this axon. Okay. Clear the screen, and here we go. So as I mentioned, I was going to mention calcium as a secondary messenger. So in this case, when the action potential has reached the axon terminal, so the end of the axon, we have to be able to send that message on. So what happens here is when that action potential reaches the end, that voltage actually stimulates calcium to enter the cell. So here we have, again, an ion channel. This one is specific to calcium, and it opens in response to that voltage. So again, here we have a voltage-gated ion channel. In this case, it's calcium's. Calcium is going to enter the cell. In this case, this secondary messenger calcium is going to stimulate neurotransmitter release. That's the response. So here we have neurotransmitter release. Those neurotransmitters will diffuse across the synaptic cleft. That's what you're seeing here. And they can come in contact with a ligand-gated ion channel. They're showing you that here. Here is a neurotransmitter already bound to a ligand-gated ion channel. And that can stimulate the postsynaptic neuron. That could be, depending on the neurotransmitter, could be excitatory and cause a depolarization, could be inhibitory and cause a hyperpolarization. But that is calcium as a secondary messenger. All right, there are disorders that are associated with channel protein malfunctions. We call them channelopathies, And these are, again, disorders associated with a malfunction of some sort. A lot of them are due to mutations causing a misshapen protein that maybe it can't fit its ion anymore. They can also be caused by autoimmune disorders. There are a lot of examples of channel pathways. I'll just go through just a couple brief ones. An example of one that affects the nervous system is epilepsy. Long and short QT syndrome is associated with the cardiovascular system. Cystic fibrosis, I'm going to go into in a little bit more detail, and that affects many systems, but probably the most prominent one is the respiratory system. The endocrine system is affected in diabetes by um, channelopathies, and myasthenia gravis is an immune disorder associated with channelopathies. So I want to go through specifically cystic fibrosis and how it's affected um, by a channelopathy. So under normal conditions, the CFTR protein is what allows chlorine ions. So again, we have an ion channel to pass. So here we have chlorine moving, and this is associated with the binding of two ATPs to each NBD unit. So here we have two NBD units, you'll notice. An ATP molecule has to be binded to each one of them, but we also have to have a phosphorylated protein here. And that is done by cyclic AMP, actually. So all three of those happen. Now this ion channel will open and chlorine can move through. And if any one of those three are not occurring, we would have a closed protein channel here. So again, this is under normal circumstances. However, if we have a channelopathy and we have a mutated CFTR protein, we can have problems, and one of those would be cystic fibrosis being impacted. So there's really two types of mutations that are associated with cystic fibrosis. There's one that's caused by a deletion of a phenylalanine amino acid at position 508, and that causes a misshapen protein. So that's the one you're seeing on the right here. We have a misshapen protein, and that misshapen protein actually doesn't allow for as good of chlorine ion transfer. This is 70% of the time in cystic fibrosis. This is the case that it's this deletion mutation. Now, cystic fibrosis is associated with heavy mucus. One of the reasons why you would get that is this chlorine transport is also associated or coupled oftentimes with water. So if that water isn't being transported, we get a really viscous, heavy mucus that the cilia in the lungs and in the trachea can't move it as effectively. And that also creates a really bad it creates a good environment actually for bacteria, but it's bad for the individual, obviously. So very pathogenic bacteria can live in that heavy viscous mucus. The other mutation that can be associated with cystic fibrosis is a lot less common, but we have that one here on the left side of our image. And that one is at position 551, and it's caused by a substitution of glycine becoming aspartame, and that's at, again, position 551 on the polypeptide. And again, it actually causes a correctly shapen protein. The problem is it doesn't allow for calcium transport, or not calcium, chlorine ion transport. The good thing is we have improved our treatment for cystic fibrosis. Several years ago, life expectancy was down around 10. That's now up around 40, actually, according to our textbook. And that is due to how we can treat this. So there is a drug called ivacaftor, also called VX770, and this drug can treat this protein and causes it to allow for chlorine ion transport. So that helps. It's not perfect, but it does help increase that protein function. So that's with that 551 mutation. On the other side, there is also a treatment, and this treatment has to do with a corrector drug. And this corrector drug has to be um, given while that protein is in the Golgi. Again, it's not perfect. We still do get a good amount of misfolded proteins, but some do end up being folded correctly um, in that Golgi from the use of the corrector drug. So there are treatments. That's a positive, but again, still a very debilitating disorder. All right, to wrap this up, we talked about signal transduction and neuronal communication. So, just to kind of talk here, we can have a mechanical stimulus. This would maybe be associated with a stretch or a pressure gated ion channel. So, that pressure, maybe that's a sensor, maybe on your skin that's sensing this pressure, and it's going to cause an action potential down that neuron. Eventually, it's going to reach an axon terminal where calcium is going to be responding to that voltage. Those ion channels are going to move open, the gated voltage gated ion channel, and calcium is going to rush in and cause neurotransmitter release, which is going to stimulate the next neuron in that chain. So, again, ion channels are a huge part of neuronal communication. They are needed in action potential, they're needed in neurotransmitter um, connection between neurons as well as they're needed in sensors for mechanical stimuli. And I hope that this lesson was helpful. And here are my references. I used a couple textbooks as well as an article summary. So take care.